All. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. I hope you had a great weekend. I hope you had a great new year. And there is so much more news. So today's topic is crucial, critical, and important. All of them mean the same thing. And I'm going to take a study from Cambridge University. I'm sure that most of you are aware of it. All of you are aware of it. I'm just going to draw it and illustrate it. The basic point, that study has so many things in it that if I go over every one of them, these all can be a lecture. I am only going to talk about one part of that study, and that is that SARS-CoV-2, or sorry, Omicron, has a new mechanism to enter a cell, and that actually makes it more efficient to enter in the cell. That makes it more efficient compared to Delta to enter in the cell. At the same time, that makes it less efficient to cause lung disease. It can cause upper respiratory disease, but not the lung disease with that much efficiency. And that actually makes the Omicron's behavior more like previous human coronaviruses from a clinical picture point of view. There still is severity of the disease that can occur, so it's not exactly like them, but it is behaving very much like it is trending in that direction. So let's look at this discussion. I think you would enjoy this one. I really believe it is good. So today there are two changes. One, I'm using OBS, so please forgive my multiple clicks here and there. And secondly, I'm using Clip Studio Paint to present this because I really wanted to draw this one. So again, on that as well, allow me to get my head around wrap my head around this. So here are the references. This is drbeen.com. This is the study from Cambridge that I'll be talking about. For this study, I have the actual study here as well, and I've highlighted. It's a huge big study, so I can't cover that all today. This is the other part of it. There are some new terms today and some new proteins we'll talk about. One of the main protein, that is cathepsin, and you would now start hearing that more and more with TMPRSS2. Just like that, you would hear less about TMPRSS2 and you would hear more about cathepsins from now on. So that is a new protein that we should be aware of. It actually behaves very much like TMPRSS2. I'll explain it. This is the green fluorescent protein. That is the GFP protein that researchers used to do this study that we'll discuss. This is the Cambridge University's news about it. All those links are in the description. This is another study. This is the mini lung, or the lung organoid. That means they have tissue that looks like lung. Some studies there. And this is another study, and that is the Malayan pangolin. We will talk about the pangolin spike protein in the discussion today as well. So just please remember that pangolin spike protein was identified from the coronavirus from pangolins in Mali. So this is that pangolin spike protein. We'll talk about that today as well. So these are the references. Now let's start our discussion. I have to learn how to keep doing this switching. But I think today's discussion is good. So let's start. So we are at the Gifts for Humanity at the Humanities Coffee Shop. Here is the first concept to keep in our mind. The concept is the following, that we know that today the coronavirus SARS-CoV-2 enters a cell by binding to ACE2 receptor. We know that. Then on the cell surface is TMPRSS2 transmembranous protease protein, a protein that can cleave or that can cut proteins. This TMPRSS2 has been instrumental for SARS-CoV-2 viruses, various variants, to cut their spike protein, which is made up of two parts, and I would discuss that. We call it priming. So TMPRSS2 is necessary for the priming of, let's say, the Delta virus. And then the virus, if you see on the right side here, then the virus fuses after the priming, the virus fuses with the cell membrane, and then that causes the RNA of the virus to enter the cell. This is Delta virus. This is how we know it. Now, in this study, there are so many interesting things. For example, this study, one study, I love these researchers. I wish I can go and handshake with them. So, number one, they found out that the neutralization by antibodies has reduced for Omicron. So, antibodies that are produced for previous variants, when they try to attack the Omicron, their effectivity is reduced. That is one they found. And that in itself is a complete topic. Second thing that they found was that the vaccine's 2 doses are almost become very ineffective. I don't want to say useless, but they have become very less effective. And they are saying that third dose is partially offering protection. This is the first time in a study I'm seeing that the words are used partial protection instead of third dose gives you the full protection, as media keeps saying. And that is the reason is researchers have to be researchers. They have to be fact-based instead of doing politics. So they simply reported that the 2 doses are almost not working. Again, this is my word, not working. They said reduced efficacy. And the third dose is offering partial efficacy. That is a very important point as well and should be discussed separately. Then they found, this is the beautiful thing, and I actually did not know this. And on Saturday, when we were doing a Zoom, there was a person from India, his name is Nabu. He brought it up and I did not know. So I said, no, this is not possible. And he then shared this study. So thanks to Nabu. So here, the third thing that is important in this study is that they found out that Omicron can enter the cell without the need for TMPRSS2, without the need for the TMPRSS2. Still needs ACE2, but does not need protease TMPRSS2. Okay, continue. They also found, because of these observations, they also found that if the virus can enter a cell without TMPRSS2, so the poor TMPRSS2 standing here saying, hey, I used to be useful, there is a problem which works in our favor. That problem is that because TMPRSS2 is not priming the virus is really not connecting on the surface. Imagine a patch. I mean, it's going to be a little intense example, but imagine I get an injury on my face and they take the skin from, let's say, my leg, and they put that on my face. You will be able to see that patch separately, correct? That is the same thing over here, that when the virus, the Delta virus, when it used to fuse with the cell membrane, there was a part of that virus like a patchwork in our cell's membrane. And that part used to be able to connect or initiate connections between multiple cells of our body, creating syncytias or multiple units made up of bigger cells. That is like if somebody grabs a few humans and tie them all together with a rope. That is a syncytia formation. So this virus, Omicron variant, cannot create syncytias with the same intensity as Delta. And syncytias are formed mainly in the lung tissue where we have the cells that have cilia on them, cells that have little rope-like structures on them, microscopic, electron microscopic little hair. And this virus kind of causes those hair to fuse with each other and tangles them up. And Omicron cannot do that. And it is a good thing in our favor. So syncytia formation is reduced. That causes less clinical intensity of the disease. That causes better disease outcome in us and less lower respiratory tract infection. So now that is it. This is the basic summary of it. If you just wanted to hear What is the talk today? The talk is basically that researchers have found that the Omicron can enter our cell without the need for TMPRSS2. This is how it has become more efficient in cell entry, and this is how it has become less efficient in causing disease in the lungs. This is why it is wiping out Delta, but also not injuring us that much. That is the summary. Now I'm going to continue to go in detail for one part of the whole study. How did they find out that there is an additional route that is built? One. And second, I'm going to discuss at the end of this that I think we are looking at SARS-CoV-3 in a good way. It's not a bad virus compared to Delta. It still is a bad virus that it can kill, but compared to Delta, it is not bad. And secondly, I think we are looking at COVID-21, not COVID-19. And I would present the case at the end of the discussion. So now let's continue with our part. Here, check this out. Now detail. The same things, but more in detail. Here is a cell. The cell has this ACE2. This is TMPRSS2 standing there. It is a protease. Protease means an enzyme that can cut proteins. Here is a SARS-CoV-2, let's say Delta. And if you see, I made its protein in two ways. One is this part, which I just marked green. Let's call it S1 part. And there is a hand attached to it. Let's call it receptor binding domain. So the receptor binding domain connects with the S2. Then the S1 is separated from S2 by our TMPRSS2, which then causes the S2 part to then fuse with our cell membrane, and that causes the remaining virus to fuse with the cell membrane as well, which then causes the RNA to enter our cell. Done? That is the normal Delta. Up to the Delta is the behavior. Now look at the right side. On the right side, TMPRSS2 is just standing idle because it is not needed anymore. And on this side of the virus, you see that ACE2 is present. The virus is actually bound to it with the same receptor binding domain, which it has changed, and it has gotten many changes on the spike protein as well. It does have S1 and S2 subunits of the spike protein. However, it does not need the TMPRSS2 to do anything. And then what will be the result of this? If this virus just stands there holding on to ACE2, then we know that whenever the ACE2 binds with its ligand means whatever binds with it, ACE2 kind of downregulates and goes in. That is behavior of this receptor. There are some receptors that stay on the surface. There are some receptors that actually detach from the surface and become soluble. ACE2 can actually become soluble as well. And then there are some receptors that when they bind with the ligand, they can actually become downregulated or endocytosed or go back inside the cell. So here, this mechanism is present. Now, next what happens is, if you see here again from the left side, this is the same diagram as I was showing before. Now what happens is, this cell has started engulfing, started drinking this virus that is attached on the outer side. There is a very, very important point that is related to ADE here. You know that I've always said that AD is not going to happen. Today, I would mention with this mechanism a possibility of how ADE can occur. Although, once again, instead of we becoming afraid of this and scared of this, Omicron clinically is actually a better virus than Delta for us. So even if it is going to possibly have ADE, that would work in our favor. So keep this part of the discussion in your mind, because this is where I'm going to connect it. So now this virus and the ACE2 are being engulfed. This is the endocytosis that is happening. When the endocytosis is complete, the virus has gone in, and inside that little envelope in which the virus is present, there is this little thing sitting in there called cathepsin. You could actually now ask me that, hey, why did you just all of a sudden magically draw a cathepsin over here? And my answer will be that cathepsins are actually present attached to the membrane everywhere. So I'm drawing something on the left side. These are all cathepsins. Then as the membrane is being invaginated, the cathepsins are present on it. It is just that they're not active. And now these cathepsins are present on the inside as well. Good. So cathepsin was present. Now the question is, how will this cathepsin become active to liberate SARS-CoV-2 or Omicron from this jail, from this phagosome or endosome? That is the basic question. We have trapped the virus. And here is why, if the virus can be liberated in this way, that from this phagosome or endosome, if the virus can get out and cause infection in the cell, that can then be mapped to antibody dependent endocytosis. That can then cause ADE as well. That means it is possible from a mechanism point of view that this virus Omicron can enter a cell as an endocytotic vesicle by ADE's mechanism and use cathepsin to get liberated and then infect the cell. But at the end of the day, that infection is beating Delta, and this is less infective for our body. So let's hope that it continues to be positive for us. Okay, so this much is good. So 1 second, I have to learn how to clear these markings I make. Next. Now the question is, why does it have less disease in the lungs? Upper respiratory system, similar disease as before. Why less in the lungs? The reason is the following, and I drew this with lots of love. So let's see if I actually did any fun thing with this. So let's say these blue ones are lung epithelium, lung surface tissue. These cells have these hair-like structures on them, cilia on them. For the medical folks here, this is pseudostratified epithelium, correct? Ciliated epithelium. So there are cilia over here. These cilia have ACE2 on them, and they have TMPRSS2 on the surface as well. So when a virus, these red ones are the viruses. When a virus attaches to the cilia or the cell surface of the epithelial cell, it fuses. These red marks are the fusion of the virus with the epithelial cell, our cell, lung tissue cell. When that happens, nearby patchy parts of the cells, they start fusing with each other. And it is actually possible that one part of the membrane from the virus fuses with one and the other part fuses with the other cell. The end result is that these cells start fusing with each other and they make unit cells. These are called syncytias or units. This little cell over here on the back and this tiny cell over here, these are the macrophages that are sitting in the, or dendritic cells that are present in the lung tissue, immune system cells. Immune system cell throw a tantrum about it. They become really intensely activated when syncytia formations occur, because syncytia formation causes the cells involved, epithelial cells, to start releasing cytokines and chemokines, and the local activation occurs. That activation causes damage to our lungs, and that causes sometimes people to die. Now, because Omicron can actually not make these patches, not fuse with our cell membranes, it can actually enter a cell without making these fusions of these nearby cells, without connecting them. This is like hotel rooms where there is door between the rooms and you can open it and two doors will be connected like a unit. Two rooms will be connected like a unit. Omicron avoids that. Because of that, Omicron can actually enter the cell, but not create fusions or that many fusions. And the result is there is less intense reaction in the lungs. That means there is less intense disease in the lungs. That is why Omicron can cause disease in the upper parts. Upper cells are usually not ciliated. Because of that, Omicron's behavior in the upper respiratory tract, the nose and the throat and this area, nasopharyngeal area, is different from the mechanism inside the lungs. Very interesting. So, Omicron does not cause intense activation of the immune system in the lungs. Continuing, which means what? It means that there is an increased transmissibility, right? Because Omicron, imagine Delta is standing and Omicron is standing, and you say, go enter this cell. And Delta is struggling with the ACE2 and asking the TMPRSS2 to say, come on, prime my spike protein, and I want to go in. Meanwhile, Mr. Omicron has already become endocytotic and gone in. I haven't yet discussed one part of it. How does that cathepsin become active? I'll discuss that in a few minutes. So increased transmissibility is one. That is how it is wiping out Delta. And there is an altered clinical picture. We have more upper respiratory tract infection. We have less intense lower respiratory tract infection. I think that slowly we'll find out that we cannot call it, or we shouldn't call it, severe acute respiratory syndrome. Maybe it has become mild acute respiratory syndrome. We'll see. So less lung damage, all in our favor. It is also actually favoring the virus as well, because virus can now live on more. Again, it has not reached that human coronavirus behavior where it doesn't say us anything. It is still killing people, but much less compared to Delta. Okay, continue. Now, how did that cathepsin become active? So let's look at this diagram. Follow my little cursor over here. So imagine that the phagocyte or endocyte has become incorporated in the cell cytoplasm. Cathepsins were connected with the membrane of that, because cathepsins are connected with the cell membrane all the time, as I mentioned before. For activation of the cathepsin, cathepsins are activated in low pH. Now, here is the delicate point. This is going to connect with ADE as well. We have in our cell lysosomes. We have done this discussion many times. Lysosomes are small microscopic or electron microscopic tiny stomachs in the cell. These are tiny pockets inside the cell that contain enzymes that can destroy proteins, RNAs, DNAs. They can destroy things. When we have a phagocyte or endocyte, when we bring in some material from outside, that material could be a bacteria or a virus, or it could even be our own receptors downregulating. It could be an antibody being brought in. Whatever it is, things can be eaten up inside the cell. When they are eaten up, then the lysosome and phagosome are connected with each other. Imagine phagosome being a cooker and lysosome being all the spices and heat that you cause in that cooker for that food to digest and cook away and melt and break. So when this lysosome binds with the phagosome, lysosome spills all the acidic environment into the, or acidic things into the phagosome or endosome, plus all the enzymes that are going to go and break the pathogen in there. However, cathepsin actually becomes active in low pH, because cathepsin's own function is also to break proteins. But this function, so normally if this was some other pathogen, it would be destroyed by all of this attack. Low pH means acidity, and then there are proteases and DNases and RNases. There are all kind of ases, the enzymes that are going to start breaking down whatever is in there. But there are some pathogens. For example, I'll give you another example, Bacillus anthracis. Bacillus anthracis can become active in the low pH and escape this jail. It uses the fire that we are giving it to kill it. Instead, it uses that to become active. Same is the situation here. As soon as the cathepsin becomes active, that cathepsin start cutting things around it, and in that process it is going to prime the spike protein, and that process that happened outside would happen here, and the virus would come out of this phagocyte or endocyte, inject the RNA in the cytoplasm. Rest is history. It's the same behavior as the others. What a mechanism. Now here, let me connect it to ADE for a second, antibodies for a second. This phagocyte, this endocyte or phagocyte here, that has come in and will bring in the whatever is attached on the surface. The same thing can happen with the antibody attached to the Fc gamma receptor on a macrophage, which is then connected, let's say, with the virus, and they both have brought in antibody, virus, or pathogen plus that receptor in an phagocytic or endocytic vacuole. Then the lysosome get attached to that. We try to acidify and kill the things inside that. But if cathepsin's usage for the Omicron is beneficial, then it is possible that Omicron would get released. And if that happens, then that infection would occur. Again, I haven't read any study that mentions it. I am only discussing the mechanisms possibility. Okay, continuing. Now, what did the researchers do? You can imagine I spent the whole day today doing this little drawing thing. Here is what they did. They took an HIV pseudovirion, meaning a fake virus that is HIV. Why did they not use Omicron itself? They said Omicron is become so weak that in the research lab, it doesn't behave fast enough to enter the cells and to show its traits. It's weak. So they said we couldn't use Omicron because of its reduced fusion capability and reduced entry capabilities or infective capabilities. So they instead did what? They took an HIV virus, empty virus, they plugged various spike proteins on it, and they created those mock viruses. What did they plug on them? What kind of spike proteins? They attached Wuhan original ancestral viruses spike protein to one group. They attached alphas spike proteins to another. They attached TMPRSS2, the deltas. You can see this is the angry one, most angry one so far. Delta spike protein to another group. Then this little cute one, the Omicron. I'm falling in love with Omicron, but it is still dangerous for us. Omicron. And they attach Omicron spike to another group of HIV viruses. Empty viruses, pseudo. It's like a car whose engine has been removed and the seats have been removed, and you just kind of push it and have the tires of various kinds. And then they also had the spike protein from Malayan pangolin that I mentioned in the beginning of this discussion. And they attached that too as well. Why did they use this Malayan pangolin spike protein? The reason is this: Malayan pangolin spike protein does not have an S1 and S2 cleavage. So even if there is TMPRSS2 present, it's not going to be able to cut it because there is no cleavage site to cut. And they wanted to use this spike protein as a control. Just like for other studies, we have humans as controls. Here we have this spike protein as a control because it cannot be affected by cathepsins or by TMPRSS2. So these were various spike proteins. In my opinion, out of this whole study, what is interesting is to keep an eye on the spike protein for Delta, for spike protein for Omicron, and spike protein for pangolin. Now let's see what did they do. Here, they also took various type of cells. So if you see these cells over here, it was not just that I wanted to draw these little scissors and knives on them. The knives are TMPRSS2 on the surface. The other little things are ACE2. So they took multiple kinds of cells. They took one type of cell here on which there was a lot of TMPRSS2 but less ACE2. There was ACE2 but not that much. Then they took another cell which only had a lot of ACE2 but almost no TMPRSS2, or TMPRSS2 was really less. Then they took another cell. I laugh at my own drawing. They took another cell where the TMPRSS2 and ACE2 were abundant on that cell. And they manufactured those cells. They changed, mutated those cells to have these configurations. Why were they doing it? They wanted to see, for example, look at this central cell. It only has ACE2 on it and almost no TMPRSS2. That means if you look at delta, if you put delta on this cell, delta needs TMPRSS2 to prime it to go in the cell for fusion. But there is no TMPRSS2 here. So if you put delta on this cell, delta will not be able to infect it. Why? Because there is no TMPRSS2 on this cell. But if there is a virus that doesn't need TMPRSS2, and you put that virus on this cell, then that virus will get into the cell and we will be able to see it in the cell. So if we can see it in the cell, we would say, well, this virus did not need a TMPRSS2. We have just proven it. So that's what they were doing. So on one end, they generated a bunch of HIV viruses with various kind of spike proteins. Every one group only showed one kind of spike protein. So they created them. Then they had a groups of set of cells with various configurations of TMPRSS2 and ACE2. Good, two. Then they did a third thing. They took two drugs, Camostat mesylate, which we talked about 1.5 years ago. Camostat mesylate is a TMPRSS2 blocker. They used that, and they used E64D, another drug that is a blocker of cathepsin. So how clever. What they're trying to now do is they're going to take various configurations of cell. They're going to put viruses on them of various spikes and see what kind of infection occurs. Then they're going to put various drugs like Camostat or E64D and see which infection was blocked. So let's do that little experiment here together. Let's go to the first one, Calu-3 cells. Calu-3 cells had lots of TMPRSS2 and good ACE2, but not a lot. And what they did was they wanted to see if the role of TMPRSS2 is important for Omicron and Delta. They had multiple cells, but let's just use two for our ease. So in one dish, they had lots of Calu cells, and they put delta on them. Delta readily went into the cell. Why? Because there were lots of TMPRSS2 to prime it, and then it fused with the membrane, went in. But Omicron did not go in with as much efficiency because there were less ACE2 and more TMPRSS2. So if it wanted TMPRSS2 to serve it, then it would have worked. But it needed more ACE2. And there were not enough ACE2. So the result was that when they saw these cells, they saw that those cells that have more TMPRSS2 on their surface, they allowed more deltas to enter, but less Omicrons to enter. Then they said, okay, very interesting. Let's go to the other set. Then they went to this human embryonic kidney cell, HEK cells. On these cells, they were more ACE2 but less TMPRSS2. Again, let's use these two sets of viruses, Delta and Omicron. This cell has ACE2s on it, doesn't have lots of TMPRSS2, doesn't have the scissors outside. You put Delta on it. What does Delta need? Delta needs TMPRSS2 to prime it. but there are not much TMPRSS2. So do you think that a lot of deltas will go into this cell? No, because they are not able to be primed. The cutting part is not there. On the other hand, they found Omicrons were happily in. Why? Because Omicrons didn't need TMPRSS2. They could just bind with ACE2, get endocytosed, activate the cathepsin by the lysosome, and then become liberated in the cytoplasm. So they said, well, very clever, Omicron. Let's confirm it. So how did they confirm it? They added E64D drug, which essentially blocks cathepsin. So now they saw that Omicron went in because Omicron did not need TMPRSS2, but it could not be liberated from the jail because E64D had blocked cathepsin. And all of a sudden, this was proven. Then they did another fun thing. The last cell, this cell's name is A549 ACE2 TMPRSS2. That is a name. What it means is that this is a cell line which is called A549. It has a lot of ACE2s on it, and it has a lot of TMPRSS2 on it. Now on this cell, if you put delta, it's going to readily get in. Why? Because lots of ACE2, lots of TMPRSS2. That's what delta needs. If you put Omicron, lots of infection would occur with that as well, because Omicron needs ACE2 at least. And inside cathepsins will become activated. So both will infect this cell readily and fast. Then what they did, this is so clever. What they did was they took these cells and they treated them with chemostat. That is, they kind of blocked the TMPRSS2 on these cells. First, they proved that these cells get infected by both very easily. Then they blocked the TMPRSS2 on them. Then they tried to infect again, and this time Omicron won and Delta failed. Why? Because the TMPRSS2 was blocked. So just a few minutes ago, or in another set, they saw that Delta was able to infect it. But now with the TMPRSS2 blocked, it is not able to. So they proved that Delta needs TMPRSS2. Then they took another set of the same cells and they treated them with E64D to block their cathepsin. And then they put Omicron on this, and Omicron could not infect. So if Omicron only needed TMPRSS2 and ACE2, then they would easily infect. But they infected with less intensity. But they could not infect as well as they were able to within the other set, where cathepsins were active. And this proved it beyond doubt that Omicron can use two pathways. Now, here's a fun thing. The endosomal pathway or endocytosed pathway has been used by other coronaviruses. It's not a new thing in it. And just like pangolin we talked, that also uses it. So this virus, in a way, is still taking features from its genera, its species. Now, was it that somebody helped this virus to behave this way? I don't know. So let's continue. Very interesting thing. So with this, there are a bunch of other parts of this study that I would discuss separately. I just wanted to focus on this one part. Now with this, if I can now present that, to me, this is a SARS-CoV-3. It could be the reason a new strain's name is given is when the strain's genotypical behavior and phenotypical behavior is very different from the previous variant's behavior or strain's behavior. So if you are within the same strain, let's say SARS-CoV-2, and the new variations continue to behave the same way, they bind with AS2, they need TMPRSS2, they cause similar infection types, then you would call them variants. But if their gene is changed a lot, if their clinical picture is changed, if their binding mechanisms are changed, if their management, their vaccine efficacy all those are changed, then that really is a new strain. Again, the new strain should not make us worried. It's a good strain. So think about it this way as well. Let's say in the books tomorrow, when this would be written, either the book will have to handle it this way, that there was a SARS-CoV-2, and when the SARS-CoV-2 reached 21's end, its behavior changed, and now it could enter the cell in two ways, and it could cause disease in a different way. The vaccines were needed were different. The efficacy of the uh, virus was different. The intensity was different. Disease outcomes were different. Management approach was different. Then how would that literature handle it? Unless they called it this was SARS-CoV-3 and this was COVID-21. And here is why. Genotype is different. Phenotype, the appearance of it, is different because it is using a different mechanism now. So that is a phenotypical behavior. Vaccines behavior is different. Pathogenesis is different. You just saw that creating the pathology, the mechanism is different. Pathology itself is different. Lower lung disease is not as intense as upper respiratory tract. Clinical picture, clinical progression is different. For example, hospital stay reduced from 8 days to 3 days. For example, most of the patient being managed in the outpatient. For example, less people needing oxygen, less people needing ICU. That is a different clinical picture. If we keep trying to shove it into SARS-CoV-2's clinical picture, then we'll have to keep saying, if this was this virus, then the clinical picture was this. Management, of course, will be different as well. For example, at some point, we might have thought that TMPRSS2 blockers can help. Bromhexins and camostat mesylate like things. Now with Omicron, we'll have to think that they may not be useful. And we'll have to figure out cathepsin blockers. Correct? Now we'll have to figure out upper respiratory tract versus lower respiratory tract. Do we have to do the same HRCTs and chest x-rays and all that, the lower respiratory tract? Do we do the prone positioning? Again, I am kind of stretching it. We still have that kind of disease occurring. The point is, behavior is changing. Management is changing. Origin, we know that the Omicron resembles more the earlier variants than Delta. And interestingly, Dr. Paul Merrick sent me a study where, I'm sure that you have read it too, where Omicron infection protects more against Delta and less against previous ones, while it looks more like previous and less like Delta. And then in this study, researchers found one more thing, and that is the previous infection is more protective than the vaccine. So the whole behavior is changing. We also know from other studies, for example, the Hong Kong University study, that 70 times more infection in the bronchi. We also know from the previous lung organoid studies that less syncytia formation, which we saw here as well. So many studies showing behavior differences. And then, of course, the protection, as I just said, it protects more for Delta, which is excellent because others are already gone. It is a Delta that it has to compete with, and fortunately, it protects against that. So that is a discussion on a new software and on a new system. So I'm going to try to now go back to this place. So thank you very much for your time. It is 44 minutes I took. I think that there are many things that are good here. We still have this virus. For example, one of my very dear friend, she is 42, Dubai. She became ill a few days ago, and had no comorbidities, and ended up desaturating and has been in the hospital for 9 days. Just before this discussion, she texted me that she is doing much better and able to start walking around. So she's recovering now. So behaviors are different, managements are different. Over there, it is interesting. Dubai, they only gave her prednisolone and blood thinners, vitamin C, and a couple of such things, but nothing else. That is not their protocol. Okay, so with this, thank you very much. Let's see very quickly. Yes, Skyfrog says, so inactivated Omicron could be an excellent vaccine. Absolutely. Absolutely. Siritha says, so you have to do me a favor. You have to actually this time share it and like it. And it's 45 minutes, so I would stop it now. Um, we can have the discussion in the chit chat. So please do me a favor, like, subscribe, and share. I know that I get the downside of long lectures, but I think you can see that all of this material was useful. So please like, subscribe, and share. And if you would like to support this work as well, there are links in the description. You can buy me a coffee, or you can become a patron, or you can use PayPal. Thank you very much. And I'm going to see you in a few minutes. If you have any questions, hold on to them. Let's talk them in the chit chat. Bye for now.